changed since I've done it. I didn't have to do any of that stuff. I had to do all that, but I didn't have to do that. An actual plan from the school. I didn't have to have a will sign. I didn't meet Will for a whole year until I was almost done. I did all that.
So you did all this? So down here, I didn't know what to put exactly.
Yeah, you'll just have to talk to Will about those dates.
And this is the form.
Yep, that's new. I didn't have to do that.
Okay.
But I think if I remember correctly, your counselor is the one that's really picky about everything.
Yeah.
Like, she's super nosy.
She's been, at least that was like her first week. No, she was pretty straightforward. Like,
I hear a lot of people complain about her.
she told me right away, she's like, I'm approving it, to let you know, so you don't have to sit around and wait.
Yeah.
I'm gonna send you an email, it's got stuff in it, it has to be done by turn in the day before.
Dude, nice that they tell you that they, mine told me about halfway through, he's like, yeah, you're good, let's just talk about what next. I love my, counselor. He is, I talked to him once a term, at the end, that's what we need to know, if I pass, here's my GPA, here's my unofficial transcript. He says, good job, and then, if there's a problem, I email him within an hour, it will be fixed. That's not normal for VA stuff.
She was good.
Tiring,
Yeah, like she gave me
rapid change of situations and all
Yeah,
the
she's just really, particular about things.
always
I don't find that.
Yeah.
That doesn't bother me.
hyper processor.
I did find it interesting, like I was overlooking that stuff, and it's like, oh, so I'm doing your job.
Periphery is just as real.
That's what I really felt.
Well,
When you get over
so they make you do that,
Mm-hmm.
so you can learn how to do this.
just
Because it's not a school thing, it's a, how to find a job, how to research.
my sister's
There's a
there,
work,
So,
which
it's
is
an employment benefit.
yeah.
and dad's doing
You just
well,
get school out of it.
as well as
Safety
can
of
be
this.
considering, but
Which was quite a lot of laptop and
Yeah.
printer and all that stuff. And anything that's required on the service,
it's pretty
that
good.
will be paid for.
My sister got there on Sunday,
And you can talk to them about it, they'll put it
early afternoon,
in the syllabus for you.
got back
Some of them will.
30 on Sunday night, so
Like I said, they bought my, $3,000 camera, they bought my, everything, all my other stuff.
yesterday we did a lot to catch up on, just
That's cool.
something else.
Yeah.
I'm excited to learn more about it, figure it out. Yeah, she was telling me, like, I'm gonna have to submit.
None of our dogs like
Is that any
very
different
much.
from the other one?
It's just like terrified. It's trying to keep babysitting,
What is it? Yeah, as soon as you submit anything monthly or whatever. Yeah, just like, like Google thing, are you school class?
You get that,
Okay.
like, once a term.
Yeah.
And then, see, this is the one that she likes, she adds
So I
things
just
to the
gave this
program.
out.
Well, so that's what she was talking about, the turn thing. So,
But you do it once a month?
yeah, or not, yeah, winter term.
Okay. And then you have to, sign book receipts. Cause you won't get a book stipend anymore,
Mm-hmm.
cause everything will be paid for, so you'll just get book at the bookstore. And then the VA just pays the bill. You just buy your books. The college sends the bill to the VA.
So does that switch maybe on, like, do I have to go anywhere or turn anything into it?
Nope. You just, sign it, send it to Will.
Saturday.
Will's pretty much our liaison.
Mm-hmm. You know, who's it's gonna be or? That's what it is.
They don't have much now.
Awesome.
The one we have that was there, now works for us. Same
Yeah, I saw a couple of them.
The last report I heard.
Just he gave me like a week.
And luckily, Bill is such a cool thing, if you're an SOU vet, I mean, I don't know if I'm supposed to talk to him or not, but he directed you in the right direction, you can go find, this department, you might not know the person. Will's a really sharp guy. Andy Gates. Fucking Bill.
Yeah, just kind of.
That's exactly how I felt with.
I use the handicap one of them 13.
Me too. I don't feel that, because I am 80% the staff.
Not feeling bad at all.
I got it.
I got up bright and early today. I was up at 4:45 and I started working on my other classes. I got like a little bit more to do on one class and then I'm done with that class.
I know, I got 3 more pictures to draw for my organism journal, and then I'm done. That's it. I don't know why I struggle so much with that.
It's all learning.
I struggled this term with like, I think because when my brother passed away at the beginning of the term, and then. Nice.
The sleep and being rested, something that I run into.
Yeah, I did 2
with
tests this
situations a
morning.
lot, too.
Started and finished at 5:30. I looked at the clock and it was 7:45.
Let's see. Where's the rest of your class?
They got lost in the heat.
Yeah.
They went looking for me.
I was gonna say, they might have freaked out because of the rain.
Oh, yeah, it was
It
raining.
rained for a good 2 minutes.
Mm.
On the way in.
gonna poke my head out before we
I
start.
walked out and jumped in my truck and I was like, what is this?
It's so weird.
It's 80 I got the windows down, it's 80 degrees.
Yeah, we're out in Williams, so we got a full, thunderclaps and like, I could be Florida for like
I heard Crescent
35
City,
seconds.
Crescent City's getting a lot of thunder and lightning right now.
Really?
Yeah.
Oh.
Did it rain tonight or this
I
morning?
can see that, that's direction it was
Felt
rolling
like
in from back
or
there.
so ago?
Okay, I was gonna say, Chris,
Yeah, it was
must be
weird.
hot.
And it's gonna be so buggy today. It already is.
Wacky.
Ugh.
Yeah, there's nobody around,
Wow.
apparently. All right, so let's at least get started. So I appreciate y'all being flexible, and hopefully this setup will work. I apologize if I'm not as smooth as I usually am. But yeah, we're in here since the labs are on their own separate AC system. So that's why we are nice and cool in here.
Is the overall system down? Is that what's happening?
Yeah,
It's busted right now.
I think the maintenance said it's like the main chiller's gone. So I guess I gotta send somebody out. So I don't know how long that'll take. It didn't sound too promising, but that's okay. We're in here, y'all are here. So we will proceed. Oh, we found it. We got more stragglers. Uh-oh. See, I knew I was gonna. And so notice, right, we're actually doing chapter 12 today. Uh-oh. Sorry. Technical difficulties. I'm having issues just seeing my mouse. There we go. Chapter 12 today. So we skipped 11. We'll come back to 11 later. So your end of the exam still goes through like 11 through 15, I think it is. But we're just doing them kind of out of order.
So this stuff won't be on the exam?
Correct.
Okay.
And so, yeah, good reminder. So this is not going to be on the exam. Did you have a question about the exam?
Oh, I did, selfishly. I'm gonna catch up with other lectures and get a good idea, 'cause it's usually class-involved things. Are we gonna do any exam review at all today, or not so much?
I probably wasn't planning on it.
Yeah, I'll be okay. I'll
Yeah,
catch up.
and we could talk privately if you need some more time or whatever. But yeah, exam 2 is coming up, so that is Thursday. I am still writing it. Chapters 6 through 10, but again, trying to go for broad picture ideas. And then right now I'll do some like map interpretation problems and stuff like that you don't have to memorize a whole book for. All right, so chapter 12, we should also remind ourselves, so we're doing chapter 12 in kind of preparation for our field trip on Saturday. And so we are meeting at 8 a.m. And I think people have the information on their paper. We can go over that more on Thursday if we need to. But again, field trip in Crescent City, 8 a.m. So probably have to leave here around 5:30 or so. So I will see you there. All right, so in preparation, we can at least talk about the marine life. And so we'll look at how, well, really all living organisms are classified by their characteristics. And we probably know, right, they're separated into different classifications, so kingdoms, phylums, all that. And so we'll look at some of their properties. You kind of got a taste for that when you dissected the fish or the shark. You're seeing all the structures, how it's sleek in the water, all the fins to help it move. And we'll look at kind of the really 2 divisions in our marine environment. And so again, DNA sequencing has clarified this genetic comparison. And so now we can kind of say, well, what constitutes life? And so we start off with, right, everything is made up of atoms. And so that's not a good distinction of what life is. And so what does something have to do, though, for it to be life? Yeah. So reproduce is the big one. So it needs to reproduce on its own by some mechanism. And then what else do living things tend to do? Eat, breathe, consume, kind of take in nutrients. So they have it here, it's capture, store, and transmit energy. But again, we see that, right? Transform nutrients either into like their shells or just take energy to move around, just like we're having energy to move around with coffee consumption and other goodies. And of course, living things also adapt to the environment over time, so they could change a little bit. Unlike, let's just say chemicals. Chemicals are always just atoms. They don't adapt. They don't do that or anything. But again, living things adapt and can even change. All right, so now let's look at kind of our main classifications. And so we start with our kingdoms. Our domains. And so this is, right, the largest box of classification. And so we have 3 main domains here. So we have bacteria, so of course just simple life forms without nuclei. Archaebacteria, so these are simple microscopic creatures, kind of more ancient organisms, at least so they think. And then everything else, including people, are eukarya, so those are any sort of complex multicellular organisms. And so in this, right, there's plants and animals, so this is fairly large. And so this is, again, just kind of doing a comparison. So if you were mapping out kind of different organisms and their evolution, so notice, right, you have relatively small amount of bacteria and archaeobacteria, and that makes sense. They're pretty small single-celled organisms. And then everything else is the eukarya.
Oh, so.
Yeah.
People would be considered eukarya.
Yep, so people, pretty much like everything we think of is eukarya. So, right, humans, sorry, handwriting.
It's similar to I don't remember if I'm saying that, but the eukaryotic organisms versus the whatever.
Yeah,
The multi-cell.
eukaryotic, so that's where it's from, exactly. So that, again, just means the multicellular. And so most organisms, right, are multicellular, at least when we think of organisms, you know. So dogs,
Because
plants.
the bacteria would be the other one, right, the prokaryotic or.
Yeah, and so the archaea, they're just like simple. So they're just even more simple than the bacteria. I think they did some sequencing to figure out where there was actually a split in the two, at least. Again, so they say.
What would be an example of that one?
Of the archaeobacteria.
Yeah.
So all those are the deep vent bacteria. So the ones, usually what characterizes the archaeobacteria is they're all like extreme, basically. So the ones, either they're in the deep sea vents or like Yellowstone and the sulfur and the acid. So any things that can survive kind of those conditions. So put really hot, maybe acidic, maybe sulfuric conditions, and not really happy conditions, but yeah, those survive there. See, anything else that we want to say? So I think that's it for our overview. And so then these are divided into our 6 kingdoms of organisms. And so we have, again, our archaeobacteria, which goes back right to our archaea domain. So that just has archaeobacteria, which, like we mentioned, were all the little microscopic. Let's see, do we have? There we go. More detailed. Right, so the archaeobacteria, like you mentioned, microscopic. And then some of them could be methane producers. They also oxidize sulfur. And so again, extreme conditions. So all these are extreme. I think some are also extreme cold. But usually we think of like the extreme, again, the thermal vents are either like hot springs, so we'll add on their Yellowstone. Oh, you're fine. And so then let's go back up. And so like we mentioned, our eubacteria, which again falls into our bacteria, are our simplest organisms. And again, the distinction with the eubacteria is they are lacking a nuclei, so they do not have a nuclei. And so cyanobacteria are included in there, so this is your like blue-green algae. And so all these, let's see, all the rest should fall under our eukarya. And so, right, we have our plantae. So now our kingdom, and of course, right, this is going to be our plants. But what's not under the plant category that we talk about in the ocean? So not in the plant category. Yeah, algae. So algae, right, algae are not plants. So algae are single cell. So they are not part of the plants, so just keep that in mind.
So what would,
So yeah, usually the eubacteria for the algae. Unless it has, so it depends if it has a nuclei, because other algae would be under the Protista category. All right, so back to categories. So plants, again, we can list, probably go crazy, right, listing plants off, so grass, trees, all that is in the plant A. Then Animalia, this is gonna be any sort of multi-celled animals. And so this is gonna have a pretty big range, so we could have our simple sponges to our complex vertebrates and humans, so wide range there, and of course we could go crazy listing examples. And so then our protista is going to be, again, now these have to have a nucleus, so kind of star that, right? The protists have a nucleus versus the eubacteria, no nuclei. And so these are going to be either single or sometimes multi-celled organisms, and so this is where a lot of our algae fall under.
Would the
Yeah.
bacteria that gets us sick be considered bacteria? Or no?
I believe so, but it also depends, because some bacteria are also viruses, so it depends if it's a virus or bacteria.
Oh.
And viruses can't reproduce on their own, and bacteria can, so that's. Let's see, other protistas. And then there are small organisms called protozoa, and so those also are under the protista kingdom there. Oh, and I almost forgot fungi. And then fungi, maybe one of the tastier ones. So mold, lichens, and of course mushrooms are all under there. So mushrooms, pretty popular. These here. I got about. And so a lot of the work, or a lot of the groundwork, was laid down by this person, so Carolus Linus. And he basically, that was his hobby, was classifying organisms and studying them. And so he came up with basically taxonomy, and that's just how you classify organisms. And of course, at the time, right, they didn't have any sort of genetic testing, so they would just go off of physical characteristics. And then later, right, things have kind of been refined, especially recently, with the genetic information, so trying to see who's more similarly genetically, and maybe that tells us about evolutionary lines. And so I think he came up with this system. I'm not quite sure if it was in its final form then, but basically, and see, now we have to add to this domain, because that was a recent addition. And so he came up this whole system, so kingdom, phylum, class, order, family, genus, and species. And all that really is, right, for our purposes, is it just breaks down the classification. And of course, right, they become more and more specific. So, right, a certain species belongs to a certain genus, and then that's encompassed in the family, and they all fit under, right, a certain kingdom. So I think we kind of know that, right, how it fans down, kind of like a family tree. You know, you could do the same thing. I thought this was a slightly weird representation of a classification, but you can tell what it's trying to say, right? The most specific is down here at the species, and where they name the dolphin. And of course, right, it falls under all these different categories. So the family, and its certain order, it's a mammal, and with chordata, and of course kingdom, Animalia. But again, we can just appreciate this. I won't make you, even though there's plenty of mnemonics, I won't make you memorize what order they go in. And so again, just kind of appreciation right now of the different levels of classification, and then kind of comparing different organisms. So we can see like a giant kelp, so that's actually a protista. And so you can see the sub-classifications there, and then comparing a bat star, so that's probably some little like sea star. Killer whale, so that's animal. And so notice, right, these are all chordatas, just like humans, so dolphins, all vertebrates, all mammals there, and of course humans are primates, so that deviates there, and dolphins and whales are cetaceans. Again, don't need to know all these details, just appreciating. And then notice, right, the last two kind of give you the name of the species, so human, Homo sapien, dolphin, Delphinus delphis. Do you guys learn Latin anymore? Do they still teach that in high school? No? No, origins of words are not important. Anyways, if you ever get bored, I highly encourage you to learn some Latin. I guess it depends if you want to, what you want to be. If you want to be a biologist, learn Latin. If you want to be a chemist, learn German. That's my, I wish I knew German. I was definitely trying to read German papers in grad school. It's not a good thing. All right, so then we'll look at kind of a few examples of some organisms. And they kind of put them in these three main categories, at least for these small organisms. So we have our plankton, and this means, right, we have floaters. And so we'll look at this in more detail, but usually they're classified by size. And so usually, right, these float through the sea. A lot of them are like extremely, extremely small. And of course, right, they filter through the animal and get consumed. And then nectin, let's see what we have. Here's a picture of an organism in its nectin stage. So basically just means it can swim now. So it may have, for example, and not all organisms go through these phases. So an organism may just be a plankton forever, and we'll look at that classification. But if it grows in its next life stage to a nectin or nectonic adult, that's what it would be, so something that can swim around. And then benthos are going to be your bottom dwellers, and so these are going to be stuck to the ocean. And so most biomass on the earth consists of plankton, which of course is pretty amazing since it's fairly small. And so the first way that we can classify them is whether they are autotrophic. And so this would probably be a good term to know, not for exam 2, but for exam 3. So autotrophic just means that you can produce your own food, any sort of organism. And so usually, right, that involves photosynthesis, especially if we're talking about plankton. And so we can also have what are called these zooplankton, and they are called heterotrophic, which just means they need to eat something else. So they can't produce their own food. So two good terms to know. And so these are nice pictures showing us kind of both size and diversity and also comparison. So here's a human hair. So this is like 100 microns. I usually say a hair is like 1 × 10⁻⁶ meters. And so, right, we've talked about some of these little plankton before. So our coccolithophores. So we mentioned these little creatures, right, and our diatoms. So all these that either take in our calcium carbonate or our silicates, right? Remember them, all those little organisms from previously. And again, just appreciate the tiny size and diversity here. So a lot of them are kind of translucent looking.
So they're really, really small.
Yeah, and so let's see if we have more detailed notes. So you're right. So they're very, very small. And notice, so we have all these different kind of classifications, all based on kind of size and also lifespan. And so looking, right, our bacterioplankton are extremely small, so on the order of bacteria, hence their name. I was trying to find, yeah, so they are 1/2 of a micrometer, 1/2 of a, You don't need to know this, but just again appreciate, right? Super small, and at least half of the ocean's photosynthetic biomass is this bacterioplankton. So that's pretty amazing. And so, right, likely our most abundant photosynthetic organism, and this is also why, so between the plankton and the algae, right, they say our ocean supplies like 50% of our oxygen or something like that. Who knows how they get these figures, but, right, basically a lot of oxygen, pretty important. And then this is a little fascinating. I wanted to look more about it, but I haven't had a chance. So this is vireoplankton, so kind of, right, virus, so think of word origins. And so again, the size just means, and so right, are you aware like viruses are smaller than bacteria? So that's, yeah, fun fact of the day, right? Viruses are usually really, really tiny, and so they're smaller than bacteria, hence why it's like also difficult to isolate and find viruses. And so these viroplankton are even smaller, and so it was only recently that they were discovered. Trying to see. Oh, they didn't have a size on there. But yeah, so from what your book says, they were kind of recently discovered, again, because they're so small, so that's kind of difficulty in trying to detect and isolate these different organisms. But that could be cool. It may also be, right, how viruses transmit through the ocean, don't really know, so probably more study needs to be done. So that's one way. So we're looking at size. And so now, maybe I should break this off a little. And so we had other classifications of size, so we'll look at this. So this is, right, basically a size comparison. And then, so these two terms just classify it as how much of their lifespan is a plankton. And so if something is a holoplankton, so I think hollow was like whole, and so that just means your whole life you are a plankton. And if you are a meroplankton, then that means you have some sort of part, so only part of your life is a plankton, so probably either, right, your juvenile or larval stages, and then you'll go into, right, your necron phase where you swim around.
So are these stages that plankton go through, or are these different types of plankton?
Different types of plankton, right? So you could have one type of plankton that's a holoplankton, and it will always be a little plankton swimming around, or if it's a meroplankton, like the squid would be classified as a meroplankton actually, because it has a larval stage. Where's our picture, right? So this is the larval stage, so it's considered plankton because it's just like small organisms that will, you know, be sucked in basically by the big whales and other fish. But then as it matures, then it becomes, right, the little swimming squid. And this was where he mentioned that it might even have a benthos life stage. Benthos just meaning stuck. So it starts off actually in the benthos stage, right, stuck as egg sacs, then moves into a plankton stage, then becomes swimming, and then goes back to reproduction. So yeah, that's just how to classify it. So now, just saying, is it always a plankton? And then again, kind of back to the size classification. And so we can have macroplankton. So even jellyfish could be classified then as our macroplankton. And so this, your book lists as 2 to 20 centimeters approximately. So that's pretty large now. Versus picoplankton, which is 0.2 to 2 microns, so pretty tiny. And so now looking at nekton, So that just, again, means our swimmers. And so this just means you have something that's an independent swimmer. And so like we mentioned, this could be either fish or squid. And then other marine mammals are considered to be nekton. So just anything that's a free-swimming organism. And so anything from little krill to the fish to the big tasty tuna to dolphins to sharks. And it mentions, right, nectonic organisms have a variety of shapes, but they're all excellent swimmers, and some even, right, can swim through long migrations. And so like we mentioned, the benthos then, and this should remind you of some word, right, with our sediment. So same thing, so referring to our bottom dwellers, our ocean dwellers. And so epifauna live on the surface of the seafloor, and infauna just mean that they would be buried into the sediment more. And we could also have nectobenthos, which swim or crawl through water above the seafloor. And then benthos, this is good to note. So benthos are most abundant in shallower water. And so, right, thinking back to kind of the coral reefs, potentially. So find a lot of life forms there. But then your others, right, may live in the darkness, coldness, and in the deep ocean. So, right, this is they're gonna have different characteristics and, right, have different adaptations to survive these different conditions.
So like an anglerfish would be one of those, or no?
I-
Do they have to actually be stuck to the bottom of the ocean floor?
Catfish.
Yeah, I think they'd have to be more stuck, but I would have to double check with that. So let's, yeah, I think like sea cucumbers would be, I'm trying to find which ones live like deep, because I think you can still get like deep sea stars. All right, back to, and so here's our little diagram of some bottom dwellers. So this is again more of like a coral reef. And so let's go ahead and label this. So we can do, what do you think one is?
A sponge.
Sea sponge, excellent. Anybody tell what 2 is?
Sand dollar.
Sand dollar. What's 3 over here? Crinoid. Anybody know what a crinoid is? I know, it looks funny. I'm going to ask Google. Let's see, what else do we have? So what's 4? Sea anemones. So we may see, right, sea anemones, hopefully, if I can spell anemones. And can anyone tell what 5 is? You drew this. Nice. Barnacles. So trying to think of what we might identify. So we should see some barnacles too. And what's 6? Mussels. I feel like our organism journals are helping. Can we tell what 7 is? Sea urchin. Excellent. To me, that's a weird picture, but maybe that's just me. And then, so let's see, what do we have? So we have 8 over here. What did you say?
Not long name.
Sea cucumber. And 9. Oh, this is a new one. Your book has sea hair.
That's weird.
Yeah, it looks weird. And then 10, I think we've, yeah, some sort of crab. And then what's 11? Sea star. Or starfish. And can anyone tell what 12 is? Abalone. And let's see. What about 13? Ghost crab. And I doubt we would see 14. Anybody know what this is? Lugworm. We can ask Google. And then 15. Oh, another worm. Annelid worm. And 16. Yummy.
Oh, okay.
All right, let's ask Wikipedia. So I looked up crinoid just to look up a little bit more about them. So they're apparently from the class Crinoidea, so that's kind of interesting. See how they're stuck on the bottom.
Are they alive or just like a?
Yeah, no, they're alive. Yeah, pretty interesting. I can't say that I've seen these out in the wild, but. And then what was the other weird one? Let's look at a lugworm. They look like worms to me, but. So apparently lugworms are kind of hard to find, so only if you go looking for them. But I guess you can see kind of their burrowing patterns in the sand.
So is that the sand that gets pushed up out of where they're digging into?
I think so, yeah. So like,
Growing up in Florida, you see those everywhere.
oh, so you seen them in Florida?
The squiggle piles.
Oh, I'm not familiar with squiggle piles, so. Anybody else seen these little piles? Maybe not on the Oregon beaches. Anyways, we won't go down the rabbit hole of where this organism lives. But okay, so that was at least a good overview of what we might see on the benthos or bottom dwellers. And so, right, kind of another classification of our benthos, right, of course, are hydrothermal vent communities. And so these are going to be more of your deep ocean benthos. And so these are usually associated, again, like we mentioned, with our hot vents. And there's probably a lot of the archaeobacteria there. Again, we mentioned those extreme conditions of heat and chemicals that those bacteria can consume. They don't have too many examples of those. And so this is kind of interesting, putting everything in perspective. So apparently we're about at 1.8 million catalog species on Earth, and that number continues to increase as we, find new organisms and classify things better, of course with genetics as well, helping us to truly distinguish species or not. And so we assume that many marine species are not yet identified due to exploration difficulties. And right, and you guys have kind of mentioned that too, is like everything that lives down there looks pretty gross. And it's only when, for some reason their bodies make their way to the shore. If they've died, maybe they come up, but pretty difficult. We mentioned just the physical difficulties of exploring the bottom of the ocean. Obviously, the pressure of all the water makes it extremely difficult to explore. And so about 2,000 new species are discovered each year. And so far there are many more land species than marine species. And so the thought with that is that the ocean has relatively uniform conditions. So we'll kind of look at that, right? It's generally the same temperature and generally, right, the same salinity. And so your organisms didn't need to really adapt a lot. And that is, again, the hypothesis. So that's why they assume there's less speciation in the ocean. And so,
That's kind of a silly assumption, right?
what?
I mean, we just got done saying that because we can't see, you know, there's a ton undiscovered yet. I mean, it's kind of a silly thing to say that, I don't know.
Yeah, I mean, I totally agree. We're kind of assuming that, so that's kind of, I guess, this is, yeah. Because there's also, it's from kind of this stat that right now we have like 87% more land species compared to marine species, but
We've
yeah.
also been able to travel and, you know, explore most of the land versus we have a very small percentage of the ocean we've actually seen.
Yeah, so like who knows? We I would say it is hard to truly know, but again, that's just kind of the speculation since land has much more different like temperatures and you know you got like rocky mountains and sandy beaches, yeah, different. So that's a speculation, but yeah, without having fully explored anything, like how can you really say for sure? And so our marine species are mainly benthic, so they're just going to be by the shore rather than pelagic. And so that makes sense too, right? They're going to be kind of in shallow areas. They also have to get, right, so they probably need some sort of sun or at least plants around them that make nutrients if they themselves do not photosynthesize. And so that leaves most of the species around the coast and everywhere, as you might imagine. And so spent some money to do a census of marine life and trying to, what is it? I'm totally blanking on the word. Where you like genetically sequence, yeah. So trying to sequence and study everything. And so they discovered 1,200 new marine species, including this little yeti crab shown here, pretty cute. And trying to assess diversity. And so again, this is that graph where we're saying there's much more different world species versus marine species. Of course, you know, we'll add what else is there to discover. And of course, you know, our understanding of animal classification gets refined again with genetic testing and then how they want to figure out where that split occurred. So trying to trace back the origins. And then of that subset of marine species, again, most of them live by the coast. And so again, our marine environment is more stable than land. We'll look at that in detail, but just talking about just temperature, different conditions. I think I'll add the term microclimates. So microclimates on land. And so this means that organisms in the ocean then are less able to withstand environmental changes, but again, the ocean isn't changing too much, so that works out. And so most of these marine animals have a lot of water in them. And so this protoplasm is just considered, right, your solution, basically, your living solution, right? So just like your body, your blood has a ton of water in it, you're made of a ton of water, and it has all these chemical reactions that take place and water going. So same thing for these organisms, except they tend to be a little bit higher percentage of water. So like this jellyfish is mainly water with just its free-flowing protoplasm. So jellyfish don't have like circulatory systems, so it's just all these like nutrients bathing around or in the protoplasm. And so all those chemical reactions, right, take place in there. And of course, right, marine animals, they're not going to dry out, so obviously they're also in the water, but that's part of being a life form, is having some sort of barrier, right, to desiccation.
What does that mean exactly, desiccation?
Drying out. So desiccate means to dry out. But right, we saw, like, for example, that shark had a nice smooth skin, so that keeps all the moisture in, just like our skin. Same thing with that jellyfish. Although I'm sure the jellyfish is a little more sensitive, right? We've all seen jellyfish with that thin membrane. But that's just it. It still has some sort of membrane to keep its own protoplasm in. And so a lot of these organisms, they have some sort of mechanism for buoyancy. So they may have one of those swim bladders or contain some sort of oil inside of them to help with their buoyancy. And so we also find different structures based on their environment. And so we find that in the cold water, they tend to have less appendages versus the warm water. And then they are also smaller in the cold. Let's see, we'll look at viscosity. I think it mentions viscosity later. And so a lot of these small organisms have what they say a high surface area to volume. And all this means is if you have a small organism, and right, you can think of the surface area, and they kind of oversimplified it with the cube, which is nice. So if you look at this one cube, it gives you its volume, so just 1 unit squared, or cubed, excuse me. And then if you take your surface area, it has 6 sides, and 6 over 1 gives us 6. So that's just a ratio. And so you just want to compare it to the big crab, and oversimplification as a cube, but comparatively, so its volume, right, 1, 2, 3, 4, so that would be 4 cubed. 4 times 4 is 16, times 4 is 64. And then do your surface to volume ratio. And so that's another factor. And so like we mentioned, we can have increased appendages to increase our surface area, so that might help. And like we mentioned, sometimes there's oil, even in our microorganisms. We saw a lot of oil in our shark. All right, so let's go back. Let's first, because we've kind of mentioned viscosity twice, but we didn't talk about what viscosity is. So viscosity, does anybody know what viscosity refers to?
It's like a thickness or like the density.
So I like thickness, but there's,
So I always think of like motor oil. It's, you know, it's, I don't know, I guess from that it would be, a more viscous thing might retain heat or, you know, transfer heat better versus something that's thinner.
it's more about like how fast it flows. What'd you say?
Hydraulic potential and surface tension.
Yeah, so it's more like consistency. So viscosity is more defined as like ability to flow. But I like your motor oil example. So let's think about motor oil, because sometimes even I get confused with viscosity. Because if we have, let's just say, right, motor oil, would it have a relatively higher or lower viscosity?
Well, it depends on what you put in.
Huh?
It depends on what you're putting in.
Like compared to water.
Depends on temperature too.
So it does depend on temperature, so we will get to that. Oh, come on, I'm trying to find just like a viscosity chart for motor oil since we're talking about it. No, I want the actual motor oil viscosity, not your 5W rating. Oh my gosh, okay. All right, so this is so let's orient ourselves. So we have viscosity, and so viscosity is measured in this weird unit of centipoises. And so just appreciate for a second, so look at your oil here, and look at how high that viscosity is. So now we're saying if something has a high viscosity, that means that it does not flow. So it's flowing, right, kind of slowly, so kind of like motor oil. And so water has a viscosity of about 1, just for comparison. And so we can look up, although I think these are different units, which is not helpful. But you can at least kind of compare relative viscosities here. This is not CPs, so that does not help us. Slightly frustrating. But here's another chart to kind of compare, again, not CP units here, but are you guys familiar with like acetone?
Nail polish remover.
Yeah, although usually when it's a nail polish remover, they like dilute it. Sometimes it could be ethyl acetate though.
They use it to remove like acrylics, like that part, gel stuff.
And so notice, right, that has a pretty low viscosity, at least compared to, so we can compare, do you know what glycerol is? It's like kind of that thick syrupy thing. And, what?
I think that's the stuff that's used in soap.
Yeah. And so you can compare just even that, right? Some sort of organic solvent flows pretty quickly. So if it's low viscosity, it means it flows fast. Where high viscosity means that it's a lot slower. So let's look at that. So motor oil, we'll just add, it was about like 3,000 centipoise viscosity. And so let's just say, right, high viscosity means that it flows slower. And so now let's look at this for water, because someone mentioned, yes, temperature dependency. And so I pulled up this chart. Let's see how that looks for y'all. So I pulled up this chart comparing, this is just the viscosity of water over a temperature range. And so notice as it gets colder, right, our viscosity increases, so it's slowing down. So, right, this goes back to the organisms and why the cold water ones only needed a few appendages. And then so going up, so looking around like 20 degrees Celsius, so that's where we have a viscosity of 1. And then it slightly decreases as we continue to go higher, and that just means it's going to flow faster. And so now going back, and so let's add something about temperature. So as temp increases, right, our viscosity, and this is where it kind of gets confusing, because our viscosity decreases, right, but that really means that it's flowing faster.
So as temperature increases, viscosity.
Yeah, viscosity decreases, which feels kind of weird because that means that it's flowing faster. Because viscosity, again, the higher number really means it's going slower.
So that's why you have, for instance, in an engine that gets really high temperatures, you want something that's more viscous so that can carry that heat, because if it's super thin, it's just gonna dissipate and you're not gonna have anything. So having that thicker
Right.
oil, it'll take that heat through the entire cooling system and recycle itself back through without getting too thin to where it's not able to actually carry the heat as the vehicle for the cooling system.
Yeah, exactly. So that's engineering at play with that.
Another really good example of something like that would be like honey.
Yeah, honey.
The colder it is, it turns into a rock. The hotter it is, it goes off the, out of the jar easier.
Yeah. I'm currently using up my honey that was previously like crystallized. It's like coming back in these hot days. Yeah, exactly. That was a good example.
You can just put it in a thing of hot water, like your whole honey jar, and it'll decrystallize and go back to running.
That takes so much time and patience and that water and its specific heat,
You're a chemist though, you like that stuff.
I like boiling. I like heating up in other substances, or just burning things. Anyways. So back to viscosity. So then, and again, right, these changes when we're talking about the water, your book, I think it's like, woo, viscosity is really changing. But it's going to be pretty minor, but still, enough to influence the organisms. We're not talking about like honey or motor oil, just water changing. But like we saw with our shark, so streamlining is often important for larger organisms, so having some sort of sleek shape. And then you also had your lateral line, so that gave it the sensory information to kind of detect salinity and other information. So all that shape. And so again, some sort of shape that offers the least resistance to fluid flow. You could even take this to Olympic swimmers or competitive swimmers. You know how they always shave their bodies and go all crazy before competition. So same sort of idea, right? Trying to be really streamlined so you have no resistance to flow through the water. And so with that, a lot of these sea creatures kind of have a flat body and some sort of tapered end. So you can kind of see that, right? They all kind of look streamlined. And obviously, right, they have fins too to kind of help with stabilization and navigation. And so of course, they can also take advantage of the water for reproduction. And so it kind of depends, right, which way it's going to. So sometimes, I think we mentioned the salmon, right? They go from the ocean to the fresh waters to reproduce. And then eels will actually reproduce in the ocean, so do the opposite. They grow up in the rivers and then go out to sea to reproduce. So it just kind of depends on their breeding pattern. But again, right, the seawater can kind of help mix the eggs and the sperm up and then distribute them.
Animals just put it all out there into the water?
Oh, yeah.
Yes, salmon, they'll dump their eggs, and then the next one will come in, they call it milking, and then it'll just dump it in the pile and run.
Yeah, and well, we saw that, I think, with our last chapter with the grunion fish, where it was a little more localized, right? They were at least taking advantage of that low tide to do that. But it's the same sort of thing, right? They just need a medium to mix together. And so, certain fish, like we mentioned with the salmon, they have specific breeding grounds, so that kind of gets concentrated. Or, in the ocean, right, it's just going to be released out with whales or tuna. And I'm sure they have, right, like areas where they typically breed, but.
Next to the page, 3 stumps down, got the 21st mile.
All right, so now we're kind of looking at our temperatures and talking about how the ocean is basically the same temperature. And this is a good picture. So looking at the open ocean, it has a pretty small temperature range. So even at the depths, it still only has a minimum of -2 degrees Celsius. And of course that goes back to, water freezes at 0, but then it goes up because it's less dense and blah, blah, blah. So all that keeps it at a pretty tight range in the ocean. And then, right, our coastal ocean, so now kind of looking at more marshes or like the Florida swamps. So now this can have, right, a greater temperature range. So now we have a max of 40 degrees Celsius. And meanwhile, right, we're probably all well aware that the land can have an even wider range. And so they list a range of -88 Celsius to a max of 58 Celsius. So that's both a pretty hot and cold range, and then also just an extremely large range. So that's where the diversity of sea creatures comes into play, and again, why they think that there's more diversity of creatures on land. And again, who knows how many more species we'll have to discover in the deep ocean, but that's at least one potential factor. And so, like we mentioned, this is kind of recap. So we have our higher heat capacity of water. That's obviously a huge dictator. And then, again, water is so beautiful. I could go on about water forever. But it freezes at 0, boils at 100, so that helps keep it regulated, right? And then our solar radiation, how it penetrates. So of course, right, the topmost layer gets the most sunshine. You have photosynthesis, and as we go down, right, it doesn't penetrate. And then of course, ocean mixing. We've seen this a lot. All of that, the current driving, salinity, blah, blah. All that, right, just keeps our entire system churning.
Is that part of like the upwelling and downwelling, just the mixing?
Yeah, so add those terms in there, right? Upwelling, downwelling, right? All those, so everything that we've talked about from the difference both in temperatures, right, but then also salinity, which we've looked at. And exactly, so we've talked about the currents too, how that helps drive the upwelling, the downwelling, just keeping everything circulating. And so again, looking at the cold versus warm water species, the floating organisms tend to be smaller in warm water. And so typically that's due to they just have a shorter life cycle. And there's also more appendages in warm water. So I think the theory is if it's moving faster, they need more appendages to help swim and stabilize, but again, not entirely sure of the theory there. And so tropical organisms, they tend to live shorter, reproduce more often, and then there tend to be more species in the warmer water as well, which we tend to see. And then, right, don't forget, we've seen this before, so there's a lot of biomass, though, in those upwelling, and so just remember, right, that refreshes our nutrients.
And that's because colder water can hold on to more nutrients and oxygen, right?
Yeah, so we have, so O2 is, there's just more dissolved at certain depths. And then, yeah, so exactly, it's going to bring in your new organisms and, right, all our plankton that we were talking about. So now your plankton can come up and, of course, get eaten. And so, again, kind of our two main classifications. So looking at temperature and then we'll look at salt. And so two differences. We can either be stenothermal, so that just means we have withstand a small variation in temperature, so narrow. And so these are going to be creatures living in the open ocean where you would not expect, right, a large temperature difference. And then if something is eurythermal, that's wide, that just means it can withstand a larger variation, and so they might live in more coastal waters where it gets a little hotter and might have more ranges, so slightly more range. And so same thing, we can also have organisms that can either withstand a small variation in salinity or other organisms that can withstand a large one. And so we can probably name some euryhaline species. So again, stenohaline, back to the narrow. So any organism that can withstand only a small variation in salinity. So again, typically found in the open ocean versus our euryhaline.
So would that be, the euryhaline, would that be anything that, so for instance, like a salmon or a steelhead that does, or do they, because their whole body composition changes, do they not qualify as?
No, that would totally qualify them, yeah, because it's because their body can change, so they are able to adapt. Yeah, so that would be an excellent example. So salmon, eel, so anything that can kind of change salinity conditions.
There's a shark that can do that too, right? Was it the bull shark?
Bro?
Swim in they can live in fresh water. That's what makes them so terrifying.
I did not, I can definitely ask Google.
My mom.
Yeah, they like brackish waters, and then a lot of times they have low visibility, like most of the world's shark bites are in Florida, and most of them are in estuaries and rivers that don't hurt us. Yeah, and they can swim up to 2 or 3 miles upstream looking for food.
That's a little scary.
Run out on the rivers and lakes a lot lately, and my mom, a couple days ago, said, you need to start watching for sharks and octopus, they're starting to come up the river.
Oh, man.
Okay, Mom,
That'd be pretty
I'll watch for that.
But you don't swim in
The
river
road is
mouths, it
dangerous
just,
enough without those.
at dawn. It's just a visibility issue, like most of the time they're gonna taste you and not eat you, and it hurts them.
Yeah, according to Google, I guess the sharks can do like really low and really high. So that's pretty cool.
Yeah, and they're dumpsters, they eat everything. So like they actually, like most sharks, when they bite a human, it's an accident because they look like a seal,
Yeah.
like surfers. But a bull shark, I guess, will actually eat anything. That's what, I guess Jaws was actually originally, written, was based on, was a bull shark, not a great white.
Oh, I didn't know that. That's cool.
The great white is just big and scarier.
Okay, I'm going to add bull shark to that list. I did not know they could go in low and high.
Now I have another, what about like, you probably know about this, Florida, was it the snakehead that can get out of the water completely? Or they can kind of slither and get something and go back, would that be the
Yeah,
same thing, they get
there's
completely out
walking
of?
catfish, walking fish,
Uh-huh.
puddle jumpers.
Would those all count, I mean, if they can be outside of water completely? I mean, there's no, there's not a high salinity and just normal.
Yeah,
That might be an obscure question, sorry.
I'm curious, yeah, because I'm not sure, because I don't see anything, I'm just looking at snakehead about like salinity differences, because I think they're just freshwater,
Are they?
at least according to our Wikipedia here, but there has to be some sort of classification of like able to be out of land or something, or some sort of descriptive.
Final
Can-
thing on shark swimming safety, if you're somewhere where you think there might be sharks or there is low visibility, don't do the overhand, the butterfly, or like slapping backstrokes. Do slow, even strokes like the frog or the dog paddle underwater because they come for sound.
Yeah, the lateral
underwater stroke slows it.
And that gives them a bigger profile too. So easier for them to see in low visibility is that shadow that you cast when you're making big movements.
All right, safety tips. And so
swim faster than your friend.
that's always a safe. I thought that was like the bear safety tip.
That's what they say. How do you swim a shark? Swim faster than your buddy.
Awful. Anyways, I would be the dead person. And so we kind of mentioned this again back a couple of chapters when we were talking about those diatoms and other organisms. And so, right, they can extract minerals from seawater, so that's part of their adaptations to these salinity conditions. And of course, right, we mentioned there's calcium carbonate in the water, or silicates in the water, and so all that as well can pass through your semi-permeable membrane.
Now, the calcium carbonate is going to be like bones and shells
Yeah,
stuff like that.
and then, right, so then you'll have little ions of calcium carbonate, or like, right, both calcium and carbonate. And then the organism takes them in, so kind of, you know, from here you have a high concentration, organism takes in some of those, and then passes out, right, water or whatever they've extracted. And yeah, so then they would use that to make, right, their bodies. So if they're alive, they're incorporating that material. Of course, if they've died, they become this material again. And so, right, all your plankton, it could be extracting. And, right, don't forget, I'll add in there our silico, flagellites. So again, just our small little microorganisms that are incorporating this into their skeleton. And where were the cocos? I've probably lost that picture now. Oh well, it's fine. And so diffusion, usually you go from a high concentration to a low concentration. So this is going to be, right, ions going in and out of the cell. And so that's showing you just how that might work. So again, you have your cell membrane that distinguishes your organism from the outside environment. And then it's showing these little nutrients. And so that's part of your cell, is they're specifically made to like pass certain ions across, you know, like sodium is favorable, potassium, all those ions. And so it can incorporate it. And then, right, the cell can, of course, push out waste, as demonstrated by the little circles. And then osmosis, right, is just in reference to water. Can't spell today. Let's try that again. And so in osmosis, water molecules move from a less concentrated solution to a more concentrated solution. And so that's shown in here. So if we have what's called a hypotonic solution, and this is just hypotonic to whatever you're comparing it to. So hypo, of course, is low. So this just means we have some sort of less ions in solution. And so if you have, and you would have to have some sort of membrane that would allow for the passage of water through. And so if they started off at equal heights, your water would move from your low salinity to your high salinity to try to make these the same concentration. And then your solution water level would increase. And so the osmotic pressure is just defined as the pressure needed to prevent this flow. And so osmotic pressure is going to, of course, increase in more concentrated solutions. That kind of makes sense. We won't go more into that. So these are good terms to know. Let me zoom out. So good terms to know. So isotonic. And in this case, they're comparing the body fluid salinity to the ocean, but this can just be in reference to any two solutions that have the same concentration. So it just depends what you're comparing it to. But again, if two solutions are isotonic, they have the same concentration of ions. And then if something is hypertonic, so of course hyper, right, is more. And so in this case, they're in reference to the organism, so saying that the organism is hypertonic. So in this case, the organism has a higher salinity than the seawater. So again, I'm just trying to clarify. So organism has more ions. And then if something is hypotonic, again, this is comparing the organism to the ocean. It's going to have a lower salinity. But again, you can compare two solutions and say one is hypotonic to the other as well. And then this is a good kind of reminder as well. So if you have two solutions that again are isotonic, so we have, right, two isotonic solutions. Don't forget, right, we have their semi-permeable membrane separating them. But in this case, if they're isotonic, there's going to be no net movement of water molecules. So our water level would stay the same. And so this is just a comparison of kind of the marine and freshwater fish and how they have adapted. So looking at the freshwater fish, so they don't actually need to drink water. And of course, right, they are hypertonic relative to their environment. And so they can absorb water through osmosis, through their skin. And then, of course, they would release some salt. And their urine is going to have a large volume of urine, so that makes sense if they're freshwater. And so, huh? A large volume of dilute urine? Yeah, well, or I don't know what I said, but yeah, they would have a large volume of urine, right? Yeah. Yeah, so they're basically always drinking. So yeah, their urine is going to be diluted. Versus, so if you compare that to a saltwater fish, so the saltwater fish is hypotonic. And so they would be drinking in large volumes of salt water and then secreting salts. They are losing water by osmosis. So they're constantly fighting that. And of course, that would mean they would have a more concentrated volume of urine then.
When they say loss of water by osmosis, do they mean like the water that they take in through their mouth is pushed back out through their skin?
Yeah, it's kind of what they're saying. So, you know, think of us sweating and evaporating. So, right, we lose water that way. And so this is more to do with the difference in concentrations, right? So the fact that your saltwater fish is comparatively hypotonic, right? So that means that in general, your water wants to leave your fish cell and go out to the ocean. Because that's how the movement of water is driven. And so, right, essentially, your fish is almost kind of fighting this, right? So that's why they're always drinking large volumes of water to replenish, but then they have to have a mechanism to secrete these salts out and, through their gills and filter that out. And so you saw this probably with the gill function, how feathery it looks. And so why it's feathery is it needs all that surface area. So of course animals extract our dissolved oxygen from the seawater through their gills. And then they do an exchange of oxygen and carbon dioxide with the seawater, again directly. And of course, if you have oxygen depletion, obviously that can kill fish, so that can lead to large areas of fish dying. And so gill structure and location can of course vary. There's some pictures of gills. Again, water in and then it goes out through the side. And there's an exchange in the little gills. And so now we go to kind of how animals survive in here and adapt color-wise. And so many marine organisms have good eyesight. That probably doesn't hold true right once you start talking about our bottom dwellers, but for the most part, we said our organisms live near the coast, so they can see well. And of course, water is transparent. And then some organisms are also transparent to try to either elude predators or stalk their prey. So of course, jellyfish are a good example of that. And so we can also see camouflage. So there's the rockfish, which we can barely see the rockfish there. And also different shading patterns. So often, right, a fish will be dark on top and kind of light on bottom. So notice that, right, the top side versus the bottom side. So quite a difference there. And so again, that just helps them kind of blend in. And so now we can get, let's look at our differences in light. And so there's a daily migration of many marine organisms to deeper, darker parts of the ocean. And a dense concentration of organisms creates a false bottom recorded on the sonar readings. So you guys remember that, right, where we had those, we saw that, yeah, with the bathymetry chapter where it was like all fuzzy. Yeah, they kind of saw like those false echo readings, and so that was indicative of a large body of life. And so, of course, right, the migration can also help with a little bit of our vertical mixing and, again, getting creatures in different areas, moving things around, so that'll help. Oh, zoomed in too much. And so, right, the daily movement could also be to get away from predators. And so it depends when the predator feeds as well, whether it's daytime, nighttime, or crepuscular, so that's twilight, which I think a lot of species hunt in the twilight.
What is that?
Twilight?
Yeah.
So kind of right when it's going from daytime to nighttime, kind of the sun setting.
Okay.
Yeah, and that's when a lot of animals tend to feed and kind of get your day dwellers. And so it just depends on the different patterns of the animals. And so sometimes these organisms in the deep sea layer only come to surface at night. And so now looking And so you would have to also, right, compare. So notice as the amount of light changes, right, this kind of changes your deep scattering layer. So at the daytime, it can go all the way down to 1,000 meters. And then now our light doesn't penetrate. And then at twilight, notice it's a little bit higher. And so that may account for more hunting if there's more mixing of species kind of going around as the light shifts. And then of course nighttime, your deep scattering layer is obviously going to be a lot higher. And then of course as the dawn comes back, our deep sea layer goes back down. And so again, this is going to influence where different creatures are going to be. So obviously, right, depends what creature we're talking about. So there's our daytime predators, and then our twilight feeders, so a lot of dolphins.
Does that have to do with maybe their eyesight or anything? Like they have to see within certain light levels to be able to track down prey? Does that have any effect on it?
Yeah, I mean, so that's part of it. And again, it depends like on like what's going on, if you're a predator, if you're a prey, and then, right, how far that light goes down means how far you can see,
So
essentially.
it's not necessarily that's their like feeding schedule, it's more like based on when they can actually see and hunt prey to the best of their ability.
Yeah, and like where the prey is too, exactly, because it's just we're kind of talking really general right now, but it depends like what it's hunting, where's the prey going to be, right? But then the prey's kind of thinking the same thing. It's like, well, I don't want to get hunted, so it's all kind of a cycle. And so that's why you kind of get this, you know, they call it a daily migration, right, of species kind of going back and forth and up and down as the light changes. And like, yeah, just like you said, it depends who's hunting who and if they're trying to get away or find food or.
So like in this picture, it's like the prey would go deeper into the ocean during the day because they're more visible, and then higher up into the ocean at night because the light.
Yeah, essentially. Yeah, and like, you know, the other main point is we're just seeing, again, the deep scattering layer, how big it is in the daytime, and then obviously it decreases with the light. So it's just, right, that's all sun intensity, how far the light goes down, all that. And so some fish use these bold colors to try to blend in. And so this might help it blend in with some coral, so disruptive coloration. So I'll just say usually with the reef. So that's where it can kind of blend in. Zebrafish as well. And a lot of tropical fish. Again, the tropical fish tend to be highly colored.
Can fish see colors?
I'm not sure. I'm sure it probably would be species dependent too.
Okay.
I know there are some because a lot of lures and stuff have been tested to have like certain colors piss them off. You know, a lot of fishing lures are meant to get a reaction out of them, and
I know like the sparkly ones like must catch their eye with specific colors.
I looked it up just now too, and it says humans are trichomats, so we have 3 cone receptors, whereas most fish are tetrachromats, having an additional cone through ultraviolet. So they can see color in ultraviolet for the most part.
Oh, I'm also seeing, because when I asked Google, mine says fish only have 2 color sensing cones, but they may be able to see in UV, but I'm not sure what their vision would be. Yeah, I'm sure there's also something funny because everything's going to look blue in the ocean because you're in the water. And I think your book Did your book show some photography, or was I just looking somewhere else? Anyways, I was looking at some pictures where they were like trying to unfilter the water to try to get like the true colors for people to see of the reefs. So I'm not sure how that kind of affects their color vision. And I mean, like I said, even your textbook doesn't have a good explanation for why the tropical fish are highly colored, but maybe they're not as vivid to each other if they can't see like the same range of color that we can.
It could be that what they're eating. I mean, because aren't flamingos pink because they eat pink shrimp? I mean,
Yeah.
it could be-
Although
Some
these
are
do
what
have
they're-
like weird patterns, but yeah, that's
It
a
says
good-
on the caption to the goby, or to the mandarin, they have bright colors and bold patterns which allow them to blend into the environment using disruptive coloration. So it's like pattern is disrupting what it actually looks like.
Yeah, I think it's because the tropical, like the coral reefs, they have erratic color, so even if they don't match perfectly, they'll still kind of blend in from predator, especially like the birds that dive in, birds have really good eyesight. And so they probably need that so they can hide from, other predators outside of the water.
yeah. And so another difficulty, so we kind of mentioned this earlier, is so we have water pressure, and of course, and this is again fascinating if you're diving, so it increases about 1 atmosphere with every 10 meters. So if you're at 20 meters, that's already 3 atmospheres on you. So that's pretty unimaginable, like 3 of our atmospheres. I can't even imagine. And so many marine organisms have no inner air pockets, and that, again, makes sense. Think of the titan sub collapsing and why you wouldn't want to have, some sort of air pocket that you would have to keep pressurized, so that would be difficult. And again, that's why the Titanic sank, because if you don't have air pockets, it's just going to sink down there and not implode. And so some of these fish also have some sort of collapsible rib cage to help them deal with these extreme pressures. And so like we mentioned, many fish have a swim bladder, so that just helps with their buoyancy. And sometimes the swim bladder could have some sort of oil in it that helps them regulate their buoyancy.
You ever had the bent one before? So when you bring a fish, when you're fishing deep water and you pull them, when I used to bass fish in tournaments, you pull them from like 60 feet deep.
Wow.
They'd come up bloated, and there's a special tool you can stick in their bladder to deflate them.
Oh, that's
Relieve the
pretty
pressure
awesome.
So that they don't just, because they'll be on their side and they won't be able to go down because you added
Yeah.
too much at once, and so they go. You have to poke them, be like right here. It vents them.
That's
Venting.
That's pretty cool.
Yeah. And you can see it like, open up their mouth and then it'll look like they're like something's like a balloon is coming out of their mouth.
Oh, that's a little creepy, but that's awesome. All right, so now we're just kind of dividing the ocean. And so like we mentioned, so I think this is important. So we have, our pelagic distinctions. So open sea versus benthic, just the sea floor. And so our pelagic environment is divided into different biozones here. Let's see. And so you have your neritic providence. So this is just from the shore. And so all water, less than 200 meters deep. So this is going to be where most of your life is. And so we can orient ourselves on this graph as well. So here's our neritic providence. So you can see, that includes our continental shelf. So right at the shoreline, so all those features. And then once you get out to the oceanic, notice that's where your continental slope starts, so where it really drops off. So that indicates our oceanic province area. And so that's just anything beyond 200 meters now. And then so our ocean provenance is further divided, and so we can look at these layers. So our top layer is epipelagic, and so this is our surface here. And so this is significant since it supports photosynthesis. So again, thinking about how much sun is coming in. And then mesopelagic region. So now that's our middle layer. So you can see, right, mesopelagic begins right underneath it. And so this can be where we see organisms of bioluminescence. And it also contains our oxygen minimum layer, which if you look at this graph, so here's right oxygen and nutrient variation with depth. And so there's right our oxygen minimum layer. Then it starts increasing again. So right at that boundary, again remembering that deep waters kind of dissolve more oxygen. Here we can also look at our nutrient maximum. It's a lot of activity there, that boundary. And so after that, our bathypelagic and abyssopelagic zones. It's 75% of the living space, so the majority of it, but of course, right, it's not going to be as inhabited. And again, probably more bioluminescence in that area. So just if you're, again, bioluminescence, able to produce light. And so you can have, right, that's where you start getting those scary looking fish, like the female anglerfish, and where it might have something to entice your predators at the depth, where they have like the little lure in their mouth. And so again, this is dominated by our light. So of course, at these depths, we don't have a lot of light. And so you can again compare our bathypelagic zone versus abyssopelagic, so very deep. Again, not a lot of light and relatively unexplored. All right, so this is a good chart. So again, just reminding us, oxygen is increasing as we go down, and we wouldn't want to have too much oxygen depletion. And again, nutrients, they tend to increase right under the mesopelagic zone there. So again, upwelling is good, bringing those nutrients back up. And so now just classifying based on light. So euphotic just means where we have enough light to exist. And so notice, right, again, that's your top layer. So going, right, your epipelagic layer where they mention is dominated by photosynthesis. And then going, so dysphotic, so notice dys, so that's telling you a small, but you still have some quantity of light. And so we can go back up to our graph here. So right now our dysphotic layer here, so in our mesopelagic region. And then after that, now it goes to aphotic, so A meaning no, photic light, so no light. And again, you can see your aphotic zone begins at your bathypelagic layer. And so then we mentioned, again, our benthic environments. And I think this is also in our chart. So supralitoral, So this is the transition from land to seafloor. And so let's look at where that is in our chart. So right there's our supra-littoral, so right where our high tide is. And then our sublittoral, so from our low tide to our continental shelf. Littoral, so now low tide to high tide.
So this is like the vertical parts of the earth itself, not the water?
Yeah, so that's like, so this is now, I know this picture is kind of odd how they oriented it, but now this is talking about our shoreline of our beach and kind of what exists there, and just dividing that out. And so we looked at those, so subneritic, so this is just your high tide shoreline to your continental shelf, and we mentioned those different zones. So that's just right, again, dividing the beach up. But this is pretty detailed. I don't expect y'all to know all these little terms. No, I don't think we should label this one. This one's a lot of terms. But just right, kind of appreciate the graph. So you have this whole subneritic region divided into your littoral and sublittoral, and that just again depends on the tides. I would say if you wanted to like go for the gold, you could try to, when you go to Crescent City, classify organisms by where you found them in these regions, but again, that's okay. We don't have to go that in depth with all this. And then again, our sub-oceanic providence, so like we've seen, again, your bathyl begins with the continental slope. I probably won't test you on this, but I would at least that's a good thing to know, right? Because we've kind of talked about this before. Again, I don't think I'll make you label it, but it's just our continental slope, like we talked about earlier, that's where you get the dipping off of our land, right? Because we're hanging out on the beach, blah, 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 everything's good, high tide, low tide. But then once we go far out and we get the drop off, that's where we get, right, our bathyal environments, and then with that, the drop off of light. So that's at least good to kind of correlate in your mind, but I don't know that I'm necessarily going to test on this. And then, of course, abysmal, it dominates a lot of the environment. And then our heddle region is even lower, and so this would only be on deep trenches. And so, sorry, I'm going to scroll back up to see if it's labeled. So right now this is deep on the sea floor, way below. And their graph is kind of confusing, I will admit. Since I think it's supposed to be like a 3D graph, but it didn't. It almost looks a little confusing. All right, I think that's mainly covering it. Any other thoughts, questions? So exam Thursday, field trip Saturday, so I will be handing back lots of papers to y'all. So you should get your organism journal. So you want to take that to the coast. And you all have had great pictures, so you can kind of compare. And again, kind of put at least this small overview together of what we're going to do at the coast. All right, I guess we'll stop here, give you guys a little extra break today. And then we'll still start lab at 11.